Neuropeptides are proteins that cells use to communicate with each other. There are 3 subunits to a neuropeptide, and one of those subunits changes for each neuropeptide family. Neuropeptide AF has an arginine and an amidated phenylalanine amino acid sequence. Different families of these neuropeptides are used for different things, like the RF-amide family, which is associated with short-term anorexia. Since neuropeptide AF is a part of the RF-amide family, the researchers wanted to find out if this neuropeptide has the same effects as others in its family. NPAF has been shown to reduce morphine's ability to block pain, inhibit insulin and somatostatin release, and lower the nociceptive threshold. The ARC projects satiety to the other nuclei from the POMC and hunger from the AGRP. The VMN and DMN deal with satiety while the LHA has to do with hunger. Chicks were used for this experiment, with the vehicle being the control. For each experiment, the chicks were all from the same hatch, and new hatches were used for each experiment.
The first experiment performed was used to determine the effects of NPAF on food and water intake and determine the blood glucose concentration of the chicks immediately following this experiment. To measure the effects of NPAF on food and water intake, different amounts of NPAF was administered to the chicks that had been fasted for 3 hours. The chicks were randomly selected to receive either 4, 8, or 16 nanomoles of NPAF with dye at the beginning of this experiment. All of the chicks had been raised the same way and had been under the same conditions of living up until this experiment. These chicks were then allowed access to food and water and observed every 30 minutes for 3 hours post-injection. Blood glucose was also taken at the end of the 3 hours. To ensure that the NPAF was the effector that was making the chicks behave differently, after the test was performed, each chick was decapitated and brain observed to see if the dye that was injected with the NPAF was present in the brain. If there was dye present, the data was used. If there was no dye present, then the results were discarded. The results of this experiment will be discussed later in this presentation. Experiment 2 was used to determine the effects of NPAF on water intake in chicks that had been restricted food. The experiment was performed the exact same way as experiment 1, except that when the chicks were fasted, they were not allowed food or water. During the time that they were observed, they were only allowed water and no food. Blood glucose was also recorded at the end of this 180-minute observation time. Experiment 3 was performed to determine if CFOS could be used as an indicator for neuronal activation. To determine this, 3 chicks were chosen at random and fasted for 180 minutes. After this, they were randomly chosen to receive either the control or 8 nanomoles of NPAF. They were then allowed food and water for 30 minutes. After the 30 minutes were up, they were anesthetized and decapitated. Immediately following this, the brain was flushed with fluids to keep it intact, and it was then placed in a stereotaxic instrument, and the brain was sectioned accordingly. The brain was then frozen and cut by a cryostat in the areas that contain the nuclei in study. The section of the brain was then washed, incubated, and stained accordingly until the reactive cells could be seen using a light microscope. 2 sections were used, counted, and then averaged together to get the value for the chicks. NPAF was used in all of these experiments, but what if it had other effects on the chicks besides hunger and thirst? This is what the 4th experiment was aimed to explain. Chicks that were used in this experiment were randomly selected to be assigned to either control or 8.0 nanomoles of NPAF. After a 180-minute fast by the chicks, they were injected and placed into an acrylic cage with food and water in opposite corners. The chicks were observed from 3 different angles for 30 minutes. Activities were measured in 5-minute intervals, and at the end of the 30 minutes, water and food consumption was recorded. The activities that were recorded were locomotion, or distance traveled, amount of time spent standing, sitting, preening, perching, or in deep rest, and the number of jumps, steps, feeding, and exploratory pecks within the food container. Now begins the results and discussion part of the presentation. For experiment 1, it was found that the chicks receiving 8.0 and 16.0 nanomoles of NPAF had reduced food intake that was consistent throughout the experiment. This is shown in Figure 1. The two dark lines in each time interval represents the 8.0 and 16.0 nanomoles of NPAF injected. At each time interval, their food consumption was significantly lower compared to the control and 4.0 nanomoles of NPAF. The 16.0 nanomole NPAF, as well as the 4.0 treated chicks, had also reduced their water intake in the later half of the experiment. Figure 2 describes these results. For the 16.0 nanomole of NPAF, there was a significant decrease in water consumption until 60 minutes into the observation, and for 4.0 nanomoles, the only significant decrease in water consumption was observed at the 90 and 120 minute intervals. There was a lessening in whole blood glucose concentration across the board for all levels of injection, but there was no diagram to show this. For experiment 2, chicks that had NPAF treatment had reduced water intake for the whole experiment. When the chicks had their food restricted, their water intake would also reduce. Figure 3 shows that there was a significant decrease in water intake throughout the observation of these chicks. Every chick that was injected with any amount of NPAF had a significant decrease in water consumption. The whole blood glucose was unaffected by NPAF in this experiment. For experiment 3, the chicks that received NPAF had an increased number of CFOS immunoreactive cells in the DM, PAMC, PAPC, and VMH with 20% or more reactivity than the control group. The ARC and LH were not affected by the NPAF injection. The figure to the left explains this by showing the number of CFOS immunoreactive cells per 0.2 millimeters squared in graph form. For experiment 4, there were many behavioral modifications associated with the addition of NPAF. Table 1 represents the activities that the chicks performed that could be counted, such as feeding pecks, exploratory pecks, jumps, escape attempts, defecations, and distance moved. Out of these, the ones that were significantly affected were the decrease in feeding pecks in the 15 to 30 minute interval, and the decrease in jumps at the 15 to 30 minute time interval. Table 2 represents the activities that the chicks performed that could be timed, such as that spent standing, sitting, deep rest, perching, and preening. Out of these time tests, the ones that were significantly affected were the increase of time in deep rest and the decrease in time spent perching. Food intake was also significantly decreased in the chicks injected with NPAF.
The anorexic threshold was suggested to be between 4 and 8 nanomoles, since the 8 nanomole chicks had reduced food intake and the 4 nanomole chicks did not. NPFF did not significantly affect food intake after 120 minutes post-injection of the neuropeptide. LP-LRF amides had anorexigenic effects lessen over 40 minutes, and those effects may be attributed to the peptide length. NPFF and NPVF didn't affect water intake, but water intake was reduced when food was taken away. The food intake was not affected by the 4 nanomole NPAF chicks, but the glucose concentration did decrease. It is possible that the NPAF could affect glucose concentrations in certain feeding conditions. There was an increase in CFOS in the DMN, PAMC, PAPC, and the VMH, which could be associated with anorexia caused by NPAF. Since these nuclei are usually associated with satiety, it was deduced that the decrease in food intake was due to an increase in satiety and not a decrease in hunger. The third experiment affected ingestion behaviors. Overall, this study showed evidence supporting the hypothesis that NPAF has a hand in anorexic behaviors as well as reducing water intake. We would keep the same experiment but have 2 groups, one male and one female. We would do this to see if there's a different effect of NPAF on males versus females. This could help with treatments for anorexia if there is a difference between genders. We would also test adult chickens versus the chicks to see if their brains react the same way to the neuropeptides. Since we know brains can grow and change as we get older and learn, we think there could be a difference in how the brains react to the neuropeptides.